So today we'll see about the sex determination in the animals and also in humans, about the sex determination and about mutation. That is about the changes that occur in DNA and about the pedigree analysis. That is how the particular characters or particular diseases are inherited from parent to progeny and how we can analyze it by means of the pedigree analysis. Okay. So first we'll see in detail about the sex determination in humans. That is generally about sex determination and also in human beings. So sex determination, actually it involves the chromosomes, right? Chromosomal determination of sex. So initially when scientists were discovering about the sex determination, that is how to distinguish between a male and a female organism, first they studied in insects, right? And then they came to the conclusion about the role of chromosomes or the genetic basis of sex determination. So what are the sex chromosomes? The sex chromosomes are also called allosomes. Okay, the other chromosomes, that is the chromosomes which are involved in the other body functions, they are called the autosomes. So there are two types of chromosomes. One is an autosome, the other one is an allosome. The chromosomes that are involved in sex determination are called the sex chromosomes or allosomes. So these include actually X and Y chromosomes. For example, if you take the human beings, there are about 46 chromosomes, right? And among these 46, that is 23 pairs, or we can say it as 23 pairs. And among these, that one pair or the two chromosomes are called the allosomes, and the other 44 or 22 pairs are called the autosomes, right? So these two chromosomes are X and Y chromosomes. So on what basis we are as a male or a female? So based on the presence of this X and Y chromosome. So autosomes are the chromosomes other than sex chromosomes. Okay. So the number of autosomes is same in males and the female. So both in male and female, the autosomes, if you take the human beings, it is only 22 pairs, both in male and female. So only thing is they differ in the X and Y chromosome. Okay. So the main chromosomes that are involved in the sex determination in human beings are X and Y chromosomes. Okay, so all the other chromosomes, that is the autosomes, is same in males and females. So how they found out is, that is scientist Henking in the year 1891, he studied spermatogenesis. So what is spermatogenesis? That is the formation of the sperms, right? The spermatogenesis in some insects. So he studied in some insects. So only in insects they first discovered, okay, and he observed that 50% of the sperm, that is 50% of the sperm, received a nuclear structure after spermatogenesis. That is only after the formation, that is after the process of spermatogenesis, they received the nuclear structures. And other 50% of the sperm, they did not receive it. So then Henking, he called this structure as the X body. That is what we are now calling it as X chromosome. He studied and he found that and he called it as X body in insects. And now it is called the X chromosome. Okay. So he studied the spermatogenesis and he named it as X body, which is called the X chromosome. So in the case, if you take different organisms, there occurs in different types of chromosomes which are involved in sex determination. Okay, so the first mechanism is XX XO mechanism, then XX XY mechanism, and third one is ZZ and ZW mechanism. So these are the different types of mechanism which are involved in sex determination. The first is XX and XO. So what is this XX and XO? Here the male is heterogametic. So what do you mean by heterogametic? That is, it shows two different types of gametes. So if you take this XX, it is called homogametic because it shows the same type of gamete, both X and X. So here, so which is the male among these two? It is heterogametic, so X. So this is the male. That is gametes with X, and another one is gametes without X. So there is no X and one is with X. So this is a male, right? And female is homogametic, that is XX, and all the gametes are with X chromosomes. All the gametes will have the X chromosomes in the case of female. So this is in the case of insects such as grasshopper. So grasshopper, it is showing the XX XO mechanism of sex determination. Okay, so it differs among different animals, right? Clear with it? Then the next type of mechanism is XXXY, right? So here also male is heterogametic, similar to that of the first one. Here also male is heterogametic, that is, but here it is, there it is XO, that is one is with X, other is without X, but here it is X and Y. Since they contain two different types of gametes, it is called heterogametic, and female is homogametic, that is it has only X, that is XX. Okay, so the examples are here in human and Drosophila. Then third type of mechanism is ZZ and ZW mechanism. So here it is different. Here if you see, the male is homogametic, that is ZZ, it is in male, but the female is heterogametic. So this is it differs from other mechanisms in that here the female is heterogametic, that is it has Z and W. So examples are in birds. So here the XXXO and XXXY mechanism, it shows male heterogamety, whereas ZZ and ZW mechanism, here it shows female heterogamety. So you're able to follow, right, what is homogamety and what is heterogamety. So here the chromosome which is having the X, that is X chromosome containing gamete, the gamete which has the X chromosome is called the gyno, that is X is called the gymnosperm, and the gamete which has the Y chromosome is called the androsperm. So it has two types of gametes. The gamete which has the X chromosome is called the gymnosperm, and the gamete which has the Y chromosome is called androsperm. Okay, so these are the three types of mechanisms which are involved in sex determination, right? So if you take the humans, that is XXXY type. So human has, we saw 23 pairs of chromosomes, and among these 23 pairs, 22 pairs of autosomes and 1 pair of sex chromosomes, right? So autosomes and allosomes. So a pair of X chromosome, that is XX, is present in female, and X and Y chromosomes are present in male. So what is happening during spermatogenesis? The male produces two types of gametes, that is 50% with X chromosome and 50% with Y chromosome. But female produce only one type of ovum with X chromosome. So there is a probability of fertilizing the ovum. So here the gametes are 50% with X, other 50% with Y, but the ovum is only with XX. So when it combines, that is when the fertilization, that is the fusion between the male and the female gamete occurs, here this ovum has the probability of mating with this X or with the Y. So 50% probability of fusion with the X or 50% of the probability of fusion with the Y. So it is only the sperm which determines whether the offspring is male or female. Okay, so which is a male and which is a female one, and this is determined only by the sperm. So they say with the that is only with the females, but actually the men determines the sex of the offspring, right? Because the sperm determines whether the offspring is male or female. So if the offspring, it takes up the X chromosome from the father, then it will be XX. Or if the offspring takes the Y chromosome from the father, it is a male. Okay, so XY means male and XX is female. So this is the sex determination in humans. That is XX and XY type. Okay, so but in the case of honeybee, that is, it is based on the number of sets of chromosomes an individual receives. For example, if you take a female, here it will be diploid. And in the case of male, it is haploid. So it is the number of sets of chromosomes. There, if you see the type of the chromosome, whether it is an X or a Y chromosome, that determines the sex. But here in the case of honeybee, the number of sets of chromosomes an individual receives, that determines the sex. Right? So in the case of fertilized egg, it develops as a female. So after fertilization, the egg is formed, and this fertilized egg, that is the egg, it fuses with the male gamete and it forms a zygote, and it develops into a female that is called the queen or a worker. But how the male is formed? It is due to the process of parthenogenesis. So without fertilization, a complete individual will be formed, and that process is called parthenogenesis. Okay, so unfertilized egg, it develops as a male or a drone, and it is called parthenogenesis. So therefore, the females are diploid. That is, they have 32 chromosomes, but the males are haploid. That is, they have only 16 chromosomes. Okay, so here they differ in the number of sets of chromosomes. And this is called the haplodiploid sex determination system. So this is another type of sex determination system, which is called the haplodiploid sex determination system, and is found in honeybees. Okay. So in this, the male produces sperms by mitosis. So here there is no meiosis, and here only the male are produced by the process that is called parthenogenesis, right? So here the males produce sperms by mitosis and not by meiosis. So this is the case of sex determination in honeybee. Okay, so we saw about the different types of sex determination, that is XX, XO, XX, XY, and Z, and ZW in different organisms. And there is another type that is called the haplodiploid sex determination system. Okay, clear? And any doubt in this? Clear? So there is another type of inheritance which is called crisscross inheritance. So what is called crisscross inheritance? Crisscross inheritance. Okay, so in the case of crisscross inheritance, it is the type of actually sex-linked inheritance. So there are several characters which are transmitted from parent to offspring through these sex-linked genes. So through these chromosomes, there also occurs a transfer of some characters. They are called the sex-linked inheritance. Okay, so this crisscross inheritance is a type of sex-linked inheritance. It is also a type of sex-linked inheritance. So here what happens is a parent passes the traits to the grandchild, passes through to the grandchild of the same sex, passes to the grandchild of the same sex through offspring of opposite sex, through offspring of opposite sex. So how the characters are transferred? For example, the father will transmit to the grandson, to the grandson of the same sex. So grandson through daughter, through the offspring of opposite sex. But the characters are transferred through daughter. Okay, so this is a crisscross inheritance, which is a type of sex-linked inheritance. So this type of inheritance is called Diagnic inheritance. And mother transmit the characters to granddaughter of the same sex, right? So it is transmitted to the granddaughter through the son. Through son. So the transmission will be son through son, and this type of inheritance is called diandric inheritance. So these are the types of crisscross inheritance, and there is another type, it is called non-crisscross inheritance. Non-crisscross inheritance. So here there will be a direct transmission of characters. For example, from father to son, and the son transmits to the grandson. So direct transmission of characters through the son and to grandson. So this is called holoandric inheritance. Then mother transmits to daughter, and from daughter to granddaughter. So this is a direct inheritance. Okay, so this is called phylogynic. Gynic, generally it represents the female. Gynecium is all right. So it represents the female. And andric, this term, it represents the male part. So here, from father to son to grandson, the transmission is called non-crisscross inheritance. It is also called holoandric inheritance, and from mother to daughter, then daughter to granddaughter, which is called phylogynic inheritance. And this is diagynic, that is the parent to grandchild of the same sex, that is from father to grandson, through the offspring of opposite sex, that is, it is through the daughter, it is diagynic, and if it is through the son, it is diandric. So this is so there are two types of inheritance. One is the crisscross inheritance, and another one is the non-crisscross inheritance. So these are all sex-linked inheritance, that they are linked with the sex chromosomes. Okay, so they are all sex-linked inheritance. So there are several diseases such as this hemophilia and then sickle cell anemia. All these are related to the sex chromosomes. So they are called sex-linked inheritance. Okay. So next we'll see about the mutation. So what is mutation? It is a sudden heritable change. So sudden, it will happen at any time, right? So it is a sudden change. And heritable change, what do you mean by heritable change? That it can be transmitted. So once the change occurs in the DNA, that is further transmitted to the offspring. So it is a sudden heritable change occurring in DNA sequence. So if there is a particular DNA sequence, and if there occurs a change in the sequence in the chromosome, the change will be transmitted to the offspring. So that is the mutation, right? It is a sudden heritable change occurring in DNA sequence that results in changes in the genotype and the phenotype of an organism. So as a result of the change in the DNA sequence, there will be a genotypic change as well as the physical expression of the phenotype. That is the phenotype of an organism. Okay, so that is the mutation. So recombination and mutation, it leads to variation in DNA. So it leads to, as a result of recombination, there will be formation of the new genes, that is new combination of genes, and mutation, it leads to change in DNA. So all these leads to variation, right? So the variation in the characteristics that occur due to recombination and mutation. So there are different types of mutation, like point mutation and frame shift mutation. So what do you mean by point mutation? That is, the mutation occurs at a particular point, right? Only in one base pair. So it is a mutation due to change in a single base pair of DNA. So for example, this is the original sequence. So all the other bases, that is TAA, you know all these, right? Thymine, adenine. So these are all the nucleic acids that are present, right? here the thymine is a base which is present here, and adenine, cytosine, and then thymine. So here what occurs in the original sequence, here this T is changed to C, right? So here thymine is changed to cytosine. So it is a change in a single base. Actually, there occurs a change only in a single base. All the other bases are the same as it is. So the point mutation is due to a change in a particular point alone. So it is due to a change in a single base pair of DNA. Example is sickle cell anemia. So because of a single base pair change, there occurs a change in the particular amino acid, right? In the case of sickle cell anemia, glutamic acid 2 valent. So there occurs a change in the amino acid. So as a result of the change in amino acid, and that leads to the defective protein synthesis, right? So that leads to a disease which is called the sickle cell anemia. So this is a point mutation as a result of a single base pair change. And what do you mean by frame shift mutation? That it is the deletion or insertion of base pairs resulting in the shifting of DNA. For example, so these are the original nucleotides that are present, right? That is A, C, G. Know that 3 nucleotides, that is, they form a genetic code, and it codes for the amino acid that is threonine arginine. So what happens as a result of deletion or insertion of a particular base? So if a particular base pair is deleted, so it leads to the defect in the reading frame. So what do you mean by the reading frame? That is, if A is deleted here, now the next 3 bases will group together. So it will become CCA, and instead of coding for the amino acid threonine, it will code for arginine. So the entire reading frame will be affected as a result of the deletion or the insertion of base pairs. So it results in the shifting of DNA. So beyond the point of the deletion, what will happen? The amino acids will change as a result of the protein that is produced will also change, right? So loss or the gain of DNA segment causes chromosomal abnormalities or aberrations. That is, there occurs a change in the chromosome structure and the number because of this loss or the gain of DNA segment. So these chromosomal aberrations are seen in cancer cells. So these chromosomal abnormalities we can see in the cancer cells. So as a result of a loss or a gain of a particular segment. So the chromosomal mutation may be either due to loss of the segment, or it may be due to the gain or the duplication, or there may be, in some cases, it may be the translocation. So translocation is the mutual exchange of the chromosomal segments between the non-homologous chromosomes. So between the chromosomes, there may be an exchange of genetic material or the chromosomal segments, right? Or it may be due to inversion. So if it is an inversion, the chromosome breaks at 2 points. So there will be nucleotides and it may break at 2 points. And then this broken piece, it rotates through 180 degree, and then it reunites in a reverse order. So this nucleotide will come over here. So it breaks at 2 points, and then it rotates through 180 degree, and then they will reunite in a reverse order. So there occurs a change of the bases which are present here in this position, right? So that is one type of a chromosomal mutation. So it may be either due to addition or deletion or translocation or inversion. Okay. But there are several agents that induce mutation. So because of the presence of these agents, they induce mutation. So what are the type of mutagens? So the agents are called mutagens, which causes mutation. So first is the physical mutagen, that is UV radiation from even sunlight, right? They may cause the cancer. So exposure to too much of UV radiations, and then alpha, beta, and gamma rays, that is, all these are called ionizing radiation. So they act as mutagens. Then X-rays. So we should not get exposed to too much of X-rays. That's why they used to say that to avoid taking X-rays during pregnancy, right? Because that may affect the fetus. So the physical mutagens are UV radiations. So UV radiations, how nowadays the UV radiations, that is because of the ozone layer depletion, right? The UV radiations are directly hitting Earth. That's why so much of exposure that in turn may act as a mutagen. So physical mutagens are UV radiations, ionizing radiations, X-rays, and chemical mutagens, mustard gas, phenol, formalin, acetic acid, formic acid, and ammonia. They act as mutagens. Okay. So clear, right? What is mutation and what are the types of mutations and what are mutagens? So the next is the pedigree analysis. So in human, control crosses are not possible. So what do you mean by this? Because we saw in pea plants that the cross can be controlled. That is what Mendel did. He took the pure tall plants and he put the pure dwarf plants. That is, for many generations they were grown as a tall and the dwarf, and he was able to control the cross, right, between the tall and the dwarf plant. But in humans it is not possible. So control crosses are not possible. So what they did, so the study of family history about inheritance is used. So about the inheritance of the particular disease or the particular trait, they use this pedigree analysis. So how they did study of family history. So about the forefathers, whether they were having the disease. So about the traits of the forefathers, and then about the grandfather, then father, then son, and then for the generations they analyzed and they made a family tree, right? So such an analysis of genetic traits in several generations of a family is called the pedigree analysis. So they made it in the form of a chart, right? So that is called the family tree or the pedigree about a particular character. So the representation or chart showing family history is called the family tree or the pedigree. So in human genetics, pedigree study is utilized to trace the inheritance of a specific trait, abnormality, or a disease. So if a particular disease is transmitted from parent to progeny, so they analyzed about the previous generation, so how they were, if any symptoms were there, and about the particular disease. So that helped to analyze about the genetic diseases. Okay, so it is used to trace the inheritance of a particular trait. So these are the symbols that are used in pedigree analysis. So there must be a common format, right, to make a pedigree or the family tree. Because if it is an universal one, so that by seeing the pedigree chart or the pedigree analysis, that everybody can analyze or they can know that about the who is infected in that particular family. Like how many males or females. So all the information should be in the family tree, right? So general common format is the squares, it represents the male, and the circles, it represents the female. So if there is a mating between a male and a female, then it is represented as a line. So male and female and with the line. So parents and children in the order of birth. So as a result of mating, here a son and a daughter. So first is son. So it is represented first and then daughter. So here son and a daughter. So parents and children in order of birth. Okay. Then dizygotic twins. So even if the twins or there is also represented, that is from the single, so two son and a daughter, right? These are monozygotic twins. So they are linked together, monozygotic twins. And then a number of children, that is 4 male and 3 female. So the number of children are represented like this. And if they are affected by the disease, it is the darkened or the blackened circles and squares. It represents the affected individuals. So here it represents the heterozygotes. That is, they act as a carriers. They are not affected, but they are the carriers for a particular disease. And if they are the carrier of the sex-linked recessive disease, it is represented by this symbol with the central dot. And this is for deceased. If a particular person is not there, like you have to put a cross. Then spontaneous abortion, it is represented as triangle. And consanguineous marriage, that is the marriages between the relatives, that is also represented in the pedigree chart. And in the sexes unknown, it is represented by this diamond. So these are the general symbols that are used in a pedigree analysis. So by means of the pedigree chart, one can find out the members in that particular family and whether they are affected by a particular genetic disease or not, right? So we can make a pedigree chart by using this symbols. Clear? So any doubts on this till now? About the sex determination, and then about the mutation, and we saw about the pedigree analysis. And in the next class, we'll see about the genetic disorders and the chromosomal disorders. Okay, so we'll discuss few questions. So how many pairs of autosomes are found in human? Whether it is 46, 23, 1 and 22. So how many pairs of autosomes are found in human? Yeah, 22 is the right answer. So they have 22. So it is asked as pairs. So you also see the question properly. Sometimes they may confuse by asking how many autosomes. So here pairs, so it is 22. So in that case, you have to write 44. So see the options and then read the question whether it is asked as pairs or simply as how many autosomes. So based on that, you answer. So here 22 is the right answer. So which of the following statements is not correct? Mutations provide variation upon which natural selection can act. The vast majority of mutations produce alleles which are dominant. Mutations arise spontaneously, infrequently, and at random. Mutation rate can be increased by artificial means. So which of the statement is a not correct statement? Yes, B is the right answer. So B is the right answer. That is, vast majority of mutations produce alleles which are dominant is the incorrect statement, but all the other statements are correct. That is, mutations provide variation upon which natural selection can act. Is the correct statement. That is, they produce variation. Mutations occur spontaneously, infrequently, and at random. So we cannot induce the mutation, right? So they occur spontaneously and at random. Mutation rate can be increased by artificial means, but it can be increased by artificial means. So these are all correct statements. Only B is the wrong statement because the vast majority of mutations produce alleles which are dominant is the wrong answer. So it is an incorrect statement. Mutation is a sudden temporary change in the organism's genetic material, change in phenotype followed by a change in genotype, change in hereditary material directed by a changing environment. It is a change in genotype which may result in new expression of a characteristics. So the correct answer here is yes, D is the right answer. So a mutation is a change in genotype which may result in a new expression of a characteristic is the right answer. So here it is not a temporary change, but it is a permanent change. So this is a wrong statement, and it is a change in phenotype followed by a change in genotype is also a wrong statement. First there occurs a change in genotype, right? And change in hereditary material directed by a changing environment. So this is also a wrong statement. So the correct option is option B. So which of the following statement about mutations is false? Mutations are source of new alleles for genes. Organisms are able to create mutations to meet their specific needs. Mutations are random events and can happen in any cell at any time. Mutation tends to be harmful or have no effect on organisms. So which of the statement is a false statement? No, it's not B. You go through the statements, no? Yes, B. So now you're able to find out, right? So B is the right answer. Because the false organisms are able to create mutations to meet the specific needs. That is the wrong statement, because it is a random process, right? We can induce it by using the mutagenic agents, that is the rate can be increased, but they are not able to create mutations to meet their specific needs. So it is a wrong statement, but all the other statements are right. So they are the source of new alleles, right? Because they lead to variation. It is a right statement. They are random events. So that is also a right statement. Mutations, some mutations actually are harmful because of the change in the chromosome structure, change in the DNA and then structure, right? So some tend to be harmful, but some mutations, they are silent mutations. That is, they have no effect on organisms, right? So this is also a correct statement. Because some, we know that in the, while studying the genetic code, we'll see in detail that is some mutations will lead to, that is, will not lead to even a change in amino acid, because there are many codons which code for the same amino acids. So single base pair change, actually, it will lead to the formation of a same amino acid. So though the mutation has occurred, it will not have any effect on the organism. So this is a correct statement, actually. Some mutations are harmful and some mutations won't have any effect on organisms. Okay. So B is the right answer. Chromosomal aberration occurs due to deletion, duplication, inversion, and translocation. So which is the right answer? Yes, B is the right answer. Both, that is all A, B, C, and D. So chromosomal aberration is the change in the structure and the number of chromosomes that occurs due to either due to deletion or duplication, inversion, or translocation. So B is the right answer. So in a pedigree analysis, circles represent males, squares represent females, solid or blackened symbol represent the individual with a recessive trait always. Mating is shown by a horizontal line connecting a male and a female symbol. So which is the correct statement here? Yes, D is the correct statement here. So D. Because square, circle, actually it represents the females, right? And squares, it represents the males. And here solid blackened symbol, it represents the individual with recessive trait. That is also a wrong answer, but it shows the affected individuals, right? So all these three are wrong statement, but the correct statement is mating is shown by a horizontal line connecting a male and a female. So this is the correct statement. Mutations can be induced with gamma radiations, infrared radiations, IAA, and ethylene. Yes, gamma radiation, that is ionizing radiations, is the right answer. Mutations can be induced with either UV radiations, IR radiations, or X-rays. And choose the incorrect option. Recombination and mutation are phenomena that leads to variations in DNA. Chromosomal aberrations are commonly observed in cancer cells. A change in a single base pair of DNA is not sufficient to cause mutation. Point mutation arises due to change in a single base pair. So which is the incorrect option among these? Yes, C is the right answer here. Because a change in a single base pair of DNA is sufficient to cause mutation. Because in the case of, even in sickle cell anemia, a single base pair change which leads to a change in the amino acid, and that is responsible for many of the symptoms. So it is sufficient to cause mutation. So this is the incorrect option. So clear, right? Maybe we'll discuss few more questions. So the first question, the nuclear structure observed by Henking in 50% of the insect sperm after spermatogenesis was. For option A, that is polar body, X body, Y chromosome, and nucleolus. So, which is the correct option here? The nuclear structure observed by Henking in 50% of the insect sperm after spermatogenesis was polar body, X body, Y chromosome, nucleolus. Yes, it is option B, that is X body. Which one of the following conditions correctly describes the manner of determining the sex in the given example? XO type of sex, that is sex chromosomes determine the male sex in grasshopper. XO condition in humans as found in Turner syndrome determines the female sex. Homozygous sex chromosomes produce male in Drosophila. Homozygous sex chromosome ZZ determine the female sex in birds. So which one of the following conditions correctly describes the manner of determining the sex in the given example? Yeah, A is the right answer. That is XO is the type of sex chromosome which determines the male sex in grasshopper. So this is the correct statement. Okay. So when released from ovary, human egg contains 1 Y chromosome, 2 X chromosomes, 1 X chromosome, and XY chromosome. When released from ovary, human egg contains 1 X chromosome. It will have 1 X chromosome. So it fuses with the another and forms an XX, right? So C is the right answer. The next one. When a mutation is limited to the substitution of one nucleotide for another, it is called translocation, point mutation, base inversion, frameshift mutation. When a mutation is limited to the substitution of one nucleotide for another, it is called translocation, point mutation, base inversion, and frameshift mutation. So here the correct option is actually option B, that is point mutation. Point mutation is the correct answer. That is when a mutation is limited to the substitution of one nucleotide for another. That is, there is a change only in one nucleotide. Okay, so that represents a point only in a single point. But if you take the translocation, it is not a single nucleotide change. And even base inversion, there's some nucleotides in between 2 points. There is a change, right? And even in frameshift mutation, what happens? There occurs a change in the reading frame after the particular nucleotide. So when there is a change in the single nucleotide, it is called a point mutation. Clear? So B is the right answer. Okay. So the first, and then the next question, select the odd one out. Violet flower, round seed, yellow pod, and tall stem. Select the odd one out. Violet flower, round seed, yellow pod, and tall stem. So the correct answer is no, it's not D. Violet flower, round seed, yellow pod, and tall stem. So which is the odd one here? So what are the dominant and recessive characters? You check in that and then answer. Among these, which is dominant, and among these, so which is the dominant and which is the recessive character? So what will be the answer? So here the answer is C, that is yellow pod. Okay, so C is the right answer. It is violet flower, round seed, and tall stem are all dominant characters. So here yellow pod is the answer for which is the odd one out among these. So C is the right answer. Okay, is it clear? So the next question is the graphical representation to calculate the probability of all genotypes of offspring in a genetic cross is called pedigree analysis, karyotype, Punnett square, and chromosome map. The graphical representation, yeah, C is the right answer. That is, Punnett square is used to calculate the probability of all genotypes of offspring in a genetic cross. Then a pure tall pea plant is crossed with pure dwarf pea plant. The progeny is self-pollinated. The ratio of true breeding tall pea plants to the true breeding dwarf pea plants shall be 2:1, 1:1, 3:1, 1:2. Yes, B is the right answer. That is true breeding tall and true breeding dwarf means homozygous, right? So that is 1:1. Then gametes are never hybrid. It is a statement of law of option A, dominance, B, random fertilization, C, independent assortment, D, segregation. Gametes never hybrid. Yes, D, because it is a statement of law of segregation. That is, gametes always will be segregated, right? They will be segregated and they will never be hybrid. So it is a statement of law of segregation. And according to law of independent assortment in a dihybrid cross, option A, there are 4 genotypes in F2. F2 contains 16 phenotypes. And option C, there is a single individual which is homozygous recessive for both the characters. And option D, it is not possible to forecast the different phenotypes. So which is the right option here? According to law of independent assortment in a dihybrid cross, option A is 4 genotypes. So in F2, actually there will not be 4 genotypes, right? So here it is given as 4 genotypes in F2. And option B, here it is in the F2 generation, 16 phenotypes. And third option, there is a single individual which is homozygous recessive for both the characters. And D, it is not possible to forecast the different phenotypes. So here the correct option is C actually. So here there are in the F2, there are not 4 genotypes. It is more than 4 genotypes, right? And there are not 16 phenotypes also. In the case of law of independent as a dihybrid cross, we saw only the 4 phenotypes, right? If you fuse the tall, for example, if you take the round and the yellow and wrinkled and green, only the 4 phenotypes will be visible in the F2 generation. So this is also a wrong statement. So the correct statement is option C, that is, there is a single individual which is homozygous recessive for both the characters, right? That is 9:3:3:1. So there is only a single individual which is homozygous recessive. So C is the right answer. Okay, clear? C is the right answer here. And the Mendelian principle which has always stood true is law of independent assortment, law of segregation, law of dominance, and all the above. The Mendelian principle which has always stood true is law of independent assortment, law of segregation, law of dominance, and all the above. Oh, the correct answer is actually B. Law of segregation is the right answer. Because why are we saying law of segregation as the true thing? Because if you take the law of dominance, it is not the true for all the cases, right? We saw in Mirabilis, that is incomplete dominance and all, law of dominance is not valid in those cases because only the particular allele will not be dominant in all the cases. So that is not true in some cases. Only in some cases the law of dominance is true. Similarly, in the case of law of independent assortment, if you see, it is also applicable only for some cases. In the case of linked genes, they always will be inherited together. So there is no separation or segregation. So that is also applicable only in limited cases. But if you take the law of segregation, that is always true in all the cases, because if you take the gametes, here they will never exist in the hybrid form and they will they always be segregated or separated. So B is the right answer. That is the Mendelian principle which has always to true is law of segregation. Okay, so is it clear, Nandini? Are you clear with this? Okay, so only law of segregation is the true, or which always stood true. Then the next question is crossing of an organism with dominant phenotype to a recessive individual is called. Crossing of an organism with dominant phenotype to a recessive individual is called test cross, back cross, reciprocal cross, and dihybrid cross. Yes, it is a test cross. A heterozygous round-seeded plant is crossed to a recessive wrinkle-seeded plant. The progeny would be option A, it is 303 rounded and 301 wrinkled, and 301 rounded with the 100 wrinkled, and 20 rounded with 99 wrinkled, and 99 rounded with 301 wrinkled. So the question is, a heterozygous round-seeded plant is crossed to a recessive wrinkled plant. Heterozygous is crossed to a recessive wrinkled plant. So the progeny would be, so what would be the progeny? So on what basis will, so these are the four, yes, A is the right answer because this is a test cross, right? So the ratio should be 1:1. So A is the right answer. Yeah. Test cross ratio in a dihybrid cross would be 1:1, 1:2:1, 1:1:1:1, 9:3:3:1. Test cross ratio in a dihybrid cross. Yeah, C is the right answer. That is 1:1:1:1. A pure tall plant is reared in a soil poor in nutrition and reached the size of a dwarf plant. If this plant is selfed, the phenotype in the F1 generation is likely to be all tall plants, 50% tall and 50% dwarf, all dwarf, and data insufficient. So what will be the right answer? A pure tall plant is reared in a soil poor in nutrition and reached the size of a dwarf plant. If this plant is self-pollinated, the phenotype in F1 generation is likely to be, yes, all tall is the right answer. Then the percentage of heterozygous individuals obtained from selfing of R, that is heterozygous individual, is 100, 75, 50, and 25. Percentage of heterozygous individual obtained from selfing of RR. So these two are crossed. So what is the percentage of heterozygous individual that you will obtain from this? Yes, 50 is the right answer. Then in rabbits, the gene for gray fur, that is G, is dominant. This is gray. This is dominant over black. In a litter, if 50% of the rabbits are gray, that is 50% the rabbits are gray, then the possible parental cross combination is option A, GG into GG, and option B, homozygous dominant into homozygous dominant, and C, it is homozygous recessive and homozygous recessive, and D, it is heterozygous, sorry, it is heterozygous with homozygous recessive. So what will be the correct option? That is, in rabbits, the gene for gray fur G is dominant over that for black fur, that is G. In a litter, if 50% of the rabbits are gray, then the possible parental cross recombination is, yes, D is the right answer because it is written 50% gray. So that is possible only with the test cross, right? With the recessive individual. Okay, D is the right answer. And in a population, a multiple allelic gene contains 5 alleles. So in the case of multiple allelism, it has the 5 alleles. Then the number of possible genotype in that population would be option A, it is 5, option B, it is 15, option C, 10, and option D, it is 18. So what will be the right answer? That is, it has 5 alleles. We saw in the case of ABO blood grouping, there are 3 alleles, right? So here the question is the number of possible genotypes in that population would be. So what would be the right answer in this? So actually here there is a formula to find out the number of genotype. It can be found out by n into n plus 1 by 2. So n is the number of alleles. So what will be the answer? So here it is given as 5 alleles. So what will be the answer here? Yes, 15 is the right answer. For example, in the case of blood group, we saw that there are 3 alleles. So what will be the answer? 3 into, here 3 plus 1, 4, right, by 2. So here it will be 12 by 2. So there will be 6 genotype in the case of ABO blood grouping. So this is the formula to find out the number of possible genotype if the number of alleles are given. Okay, n into n plus 1 by 2. So this is the formula. Okay, clear? Then out of a population of 800 individuals in an F2 generation of a cross between yellow round and green wrinkle pea plants, what would be the number of yellow and wrinkle seeds? What would be the number of yellow and wrinkle seeds? So the answers are option A 800, option B 400, option C it is 200, option D it is 150. So you're able to follow the question. In a population of, that is, there are totally 800, okay, in a F2 generation of a cross between yellow round and green wrinkled, of yellow round and green wrinkle. What would be the number of yellow wrinkled? It is asked, what would be the number of yellow wrinkle? So in the ratio, if you see the dihybrid cross ratio, it is 9:3:3:1. So this 9 is yellow round and 1 will be wrinkled green. So actually this 3 will be, one is yellow wrinkle and the other one is green round, right? This is yellow wrinkled and another one will be green round. Okay, so out of these 16, if you calculate 3 or the yellow wrinkle, now you calculate. They asked how many will be yellow wrinkle. So out of the 800, so what will be the proportion of yellow wrinkle? 200. How 200 it will come? 200 is, you are saying 1/4 of the thing, right? So it is not 200. So what will be the thing? Out of 16 parts, that is, if you calculate this, out of 16 parts, only 3 are yellow wrinkle. So you have to calculate as per that. So if this is equivalent to 16 parts, only 3 parts of it is yellow wrinkle. I hope you are able to follow, right? So out of this, if a 16 parts is equivalent to 800 individuals, only 3 parts are yellow wrinkle. So what will be the ratio? First you calculate it for 1 part. So what will be the 1 part? If for 16 parts it is 800, and what will be for 1 part? Then you calculate for 3. So what will be the answer? So for 1 part it is 50, right? For 16 parts it is 800. For 1 part it will be 50. So for 3 parts it is 150 is the actually correct answer. So on what basis you put 200? So how did you put it as 200? So are you clear now? Clear with this ratio, that is 9:3:3:1. So are you able to follow it, Nandini, this sum? Okay. So you be clear with this ratio. So in the case of diabric cross, this will be the dominant character, that is yellow round, and this is yellow wrinkle, and this is green round, and this is green and wrinkle. So we have to find out for this 3. So the value will be 150. Okay. The next question is a blue fowl which is obtained from mating between black and white fowls is cell cross. The F2 ratio is 1 black is to 2 white is to 1 blue, and option B 2 black is to 1 white is to 1 blue, and option C 1 black is to 2 blue is to 1 white, and none of the above. That is, when a blue fowl is obtained from mating between black and white, okay, we get blue. The F2 ratio it is asked is 1 black is to 2 white is to 1 blue, and here it is 2 black is to 1 white is to 1 blue, and then third one 1 black is to 2 blue to 1 white, and none of the above. So which is the right answer for this? Yes, C is the right answer. So though you know that the ratio is 1:2:1, and you have to be clear which one is 1, which one is 2, which one is 1. Okay, so C is the right answer. So I hope you are clear with it, right? So practice more problems related to this genetics, right? Calculation. So mostly those type of concepts you have to be clear with this in this chapter, that is principles of inheritance and variations. So only the one topic that is left out is Mendelian disorders and the chromosomal disorders that we'll see on Monday. Okay, so with that we'll be completing this chapter. So focus more on the problem related questions, and about the ratios you should be clear. Okay. Want to have questions? What type of questions for practicing these problem oriented? Yeah, sure. And you can also practice in the app also. Maybe I'll send some questions also. Yeah. Mostly these problem oriented questions I'll send in the group. Yeah, sure. You can practice, and maybe you can post the answers and we can check. And if you have any doubts, we can discuss in the class also. Okay. Okay, any doubts in this till now? Okay, then so we can wind up, and I'll send some questions. Yeah. Okay, thank you.